So, good evening, everyone. Am I audible to you? Am I audible? Can you just give me thumbs up if you can really see my screen and can hear me? Am I audible? Can you just give me thumbs up if you can? So, how are you all? Hey, Dr. Pranay. Hey, Shubham. Perfect. Good evening, Abhishek. You are from Mysore. That's why you have kept the name Abhishek Mysore. Okay, in this session, I have already like few questions from the students. Okay, so I'll start with those questions, and then as the time will go, you can just keep asking the question in the chat box, right? I'll try my best कि अभी सभी हो जाए, but if not, then I'll answer them in the Telegram group. Okay. So usually I get so many questions related to the MODI, right? And this is one of the question a student asked me about. Like he gave me this question about a lady who is pregnant, right? Running through the second trimester of the pregnancy, right? And she passed the OTT, like the difference between the normal glucose tolerance test and this was very like not a huge difference was found, right? So doctors figured out she was suffering from MODY2, a mutation of which gene is responsible for MODY2. Anyone anyone who can tell me what could be the answer for this, right? We know the insulin dependent issues, the diabetes, we have type 1 diabetes mellitus, type 2 diabetes mellitus, right? Gestational diabetes is also there, and the MODY2 is there, right? So what could be the answer for this? Which gene is responsible, and what enzyme is related to that gene which is responsible for the MODY? Can anybody tell me? Enzyme is glucokinase, right? Enzyme involved is glucokinase, right? What kind of a MODY is? MODY is an autosomal dominant in nature, and GCK gene is responsible for the MODY. If there is a mutation in this GCK gene, what GCK gene does is it is responsible for the synthesis of enzyme glucokinase, right? Glucokinase is the enzyme which converts glucose into glucose 6 phosphate, and this enzyme also triggers the release of insulin, the release of the insulin from the pancreatic beta cells. So if there is any defect in this gene, right, any defect in this gene, then the glucokinase won't be made properly, right? And this gene is located on the chromosome 7, and that is going to hamper the insulin release. So it is like not it is not completely cutting the insulin production down. What it does, it you just need to have more insulin, or glucose level, blood glucose level should be increased way higher, only then the insulin will be released, usually under normal circumstances, right? Under the normal circumstances, the insulin is released at 5 when the blood glucose is 5 millimolar, right? At this point in time, insulin is released, but because of the mutation in MODY2, when the blood glucose concentration goes up approximately 7 to 8 millimolar, at that point in time insulin will be released. So there will be kind of a hyperglycemic condition all the time, right? That is because of the mutation in the gene, right? There is the normal ability, right? If they say that the insulin is not at all being made, that is untrue, that is a false statement. There is a normal ability, it is just less responsive in nature. It is just less responsive in nature. There will be like mild increase in blood sugar, and that is why it is asymptomatic. And when we usually diagnose it, it is detected accidentally. Maybe a patient went right for some kind of a test and they figure it out, okay, the blood glucose is high. That is accidental knowing of. And also in the majorly during the pregnancy, right? OGTT is much less abnormal as compared to impaired fasting blood glucose level. And how do we manage it? By giving the Brandon. Okay, so usually this is a question related to MODY 2. There are other relevant questions from your OBGYN also, like that is related to the head size of the baby when the mother is suffering from the MODY 2. So definitely you may ask Dr. Ramaichi for that. Okay. Now there was this confusion always about the pasture and the Crabtree effect. I don't know why many of you have like issues with these effects, and you know it very well. What is Crabtree effect? Crabtree effect says the aerobic glycolysis, right? The normal glycolysis that is happening in the body is inhibited upon increasing the oxygen supply, right? And how you are going to memorize it, it's very, very easy if you understand, if you go back to your high school and try to understand it. Pasture effect पढ़ा था सबने, ठीक है, स्कूल में, school times में everybody did the Pasture effect. What did Pasture said? He was talking about the yeast and the fermentation. ठीक है, अल्कोहल का बनाने का प्रोसेस, he figured it out how we make the alcohol. He said that yeast क्या करता है? Yeast takes the pyruvate we are getting from the glucose, ठीक है, वो तो हम we can correlate. Pyruvate in the absence of oxygen, right, and yeast is present, yeast is going to ferment it to produce the ethanol, and the carbon dioxide. Pasteur accidentally figured it out कि alcohol कैसे बनाया जाता है, right? And under what condition? When there is no oxygen available, right? After some time, he said that when oxygen is available, right, the same yeast will convert pyruvate, right, and this pyruvate will undergo the TCA cycle. What do we mean by that? That is going to generate the energy. When this is happening? When the oxygen is present, in the presence of the oxygen. This is going crazy, I don't know why. Okay, in the presence of oxygen. So there are two case scenarios. Oxygen hai to means yeast is going to multiply. Yeast grow करेगा, multiply करेगा, energy चाहिएगा for their own production. And when oxygen is not there, it is going to give you the ethanol. That was Pasteur effect. But Crabtree effect, C. Crabtree, was explained by a scientist when he was studying the cancer cells. He was not talking about the yeast, but he saw a phenomena which was very similar to the yeast. That cancer cells को क्या चाहिए? They have to grow they have to divide very rapidly. So they need lots of glucose. बहुत glucose consume करते हैं, right? And that glucose is converted into pyruvate, right? And that pyruvate, right, Crabtree said that pyruvate is going to undergo kind of a fermentation kind of a process, that is anaerobic glycolysis jayega, right? Similar to the one yeast did, right, and produced the ethanol. Similar kind of phenomena happen in the cancer cells, right? In the in the presence of oxygen. But when the oxygen supply is being increased, right, then the aerobic glycolysis is inhibited. That is particularly for the cancer cells, and that is figured out by the scientist named Crab, so hence the name Crabtree effect. Cancer cells are not looking for the ATP. Majorly they derive the ATP from the substrate level phosphorylation reaction. They need all these pyruvate ethanol thing happening because there are so many intermediates जो बीच में बने हैं. They consume it for the division purpose. They utilize those substrates for the division of the cell. Okay, so are we good about the Pasteur and Crabtree effect? I'm pretty sure after this you won't be able, like, you should be able to attempt all the questions in the exam. Now, a polyol pathway. Polyol pathway I want to discuss. Why I want to discuss polyol pathway is diabetic patient usually suffer from the cataract. Why is that? Under normal circumstances, what do we know that glucose fed state में क्या होता है? Glucose either will go under glycolysis, right? Glycolysis होगा, glycolysis will go to the TCA cycle and it will provide you the ATP, right? Extra glucose will undergo glycogenesis. We will store the glycogen, right? We will store the glucose in the form of the glycogen in the liver, right, and in the muscle cells so that we can utilize it later, right? What else happens? A part of it, approximately 5% of this glucose undergoes polyol pathway. There is another major thing that is happening, HMP, shunt also, right? But a very, very tiny amount undergoes the polyol pathway. That is when we are in the normal state. कोई diabetes नहीं है, कोई कुछ नहीं है. But what we know in the diabetic patient, right, since there is insulin release is hampered, right? Insulin release hampered होगा, so these major pathways, they all will be blocked. What we have high level of glucose in the body. So how the system body takes care of the glucose, it reroutes now this 5% to now 90% towards the polyol pathway. And the major enzyme of this polyol pathway is aldol reductase, right? And aldol reductase is majorly found in lens, retina, Schwann cells of peripheral nerves, placenta, RBCs. Majorly it is found there. And what this aldol reductase does is it converts your glucose, the excess glucose which is not taken care by the system because of the absence of the insulin. It will convert your D-glucose into what? I'll humbly request you to ask a relevant question. This is a biochemistry session, right? So glucose will be converted into sorbitol, right, by the enzyme aldol reductase. By the enzyme aldol reductase, okay? And this sorbitol is osmotically active. This is osmotically active. It is going to suck in the water, right, from the surrounding cells, and that is going to cause the cataract. Okay, so this is why the majorly, like in the diabetic patient who is responsible for the cataract, aldol reductase is responsible for the cataract. Are we clear with this? ठीक है. Do we have many more questions? Okay. Okay, I'll take that. Now, many a times we have issue with the glycogen, understanding all the glycogen synthesis, right? When it is happening. We know whenever we eat something, we go through first step is the fed state, then the fasting state, then the prolonged like starvation, then the prolonged starvation. During the fed state, there is abundance of glucose, right? Glucose is present in abundance and which we store in the form of glycogen, which we store in the form of glycogen. Majorly where? In the liver. Liver is the major site of the glycogen, right? And also it is stored by the muscles as well, right? So that it can be utilized during the fasting condition. So question is usually many a times we are confused who is triggering the glycogen synthesis. Glycogen synthesis is glycogenesis. Genesis, synthesize कर रहे हैं हम glycogen का. Who is triggering that? Insulin. That is in the fed state. ठीक है. Opposite of this happens in the fasting state. Opposite of this is happening in the fasting state. Fasting state में what happens is we are doing glycogenolysis. Glucose is not available. To replenish the blood glucose level, we have to provide the glucose, and that is coming from the breaking down of the glycogen, right? And who is triggering this? Glucagon. Now we have two processes, glycogenesis, that is a synthesis, and that is taken care by insulin. Now we have glycogenolysis, that is breakdown, that is a catabolic pathway, and that is triggered by glucagon, which is a catabolic hormone. They work opposite to each other, right? And but I told you that liver and the muscles, they both store glycogen, right? So glucagon is going to trigger liver for the glycogenolysis. Who is triggering the muscles for the glycogenolysis? It is not the glucagon, but what? Epinephrine. Epinephrine is going to trigger. That is one thing. Who else does it? Your calcium levels. Because during the vigorous exercise, calcium during the vigorous exercises, loss of calcium has been released. That also triggers the breakdown because ATP is needed, right? So there is a reduced level of ATPs. That is we have to replenish the ATP stores, and the calcium, increased calcium levels also triggers the muscles for the glycogenolysis. These two things. Okay, Satvik is asking, when will you make a video for scope of biochemistry for newly passed NEET PG aspirants? Satvik, I can do it soon. As soon as I'll get time, I'll make a video, right? You should expect it soon. Okay, now, so first of all, we should be knowing who is doing this. Insulin is glycogenesis, and glucagon is glycogenolysis. And what are the cascade of events? Do we know the enzymes which are involved for the glycogen synthesis? Which is the rate limiting enzyme? Glycogen synthesis. For the synthesis we need the synthase, right? And for the glycogenolysis, what we need? There is enzyme glycogen phosphorylase, right? So for liver you have specific glycogen phosphorylase, that is liver glycogen phosphorylase, and for the muscles you have skeletal muscle glycogen phosphorylase. You have two different glycogen phosphorylase for the different tissues. Who is triggering it? You have to remember the cascade of events. So let's talk about how it is happening. Epinephrine. Can glucagon trigger the glycogenolysis in the skeletal muscles? No. Skeletal muscles, they do not have the receptors for the glucagon. They have the receptors for the epinephrine, right? So epinephrine is going to do this in the muscles, whereas glucagon will do it in liver. Is going to do it in liver. How it is done? Actually, once there is this membrane, cellular membrane, it has what? It has receptors, the glucagon receptors to it, right? These glucagon receptors, they activate what? Adenylate cyclase. Name is itself indicative. Adenylate, that is coming from adenine, that is A. AMP, ATP, ADP has A as a base, that is adenine. So it is going to cyclize that. It is going to cyclize what? ATP. ATP gets converted into cyclic AMP, right? So during the fasting state, glucagon is, first thing is, glucagon is increasing the level of cyclic AMP. That is one thing you have to know it, right? That is increasing the level of the cyclic AMP. This cyclic AMP actually further can be converted into 5-AMP, right? That is another trigger point for the glycogenolysis in the muscles. So 5-AMP levels are up in the muscles during the starvation state, right? Or during the fasting state. Remember this. This is another importantly asked question. 5'-AMP and 5'-AMP are synthesized from the 5-AMP. Because during the starvation, do you have ATP or ADP? ADP levels are very, very high, right? So ADP combines with ADP, right? Because there are so many ADPs available, they make 5'-AMP. So these levels, 5'-AMP levels, are up during the fasting conditions, and that triggers the glycogen, phosphorylase for the breakdown of the glycogen stores in the muscles. Okay, now let's coming back to this. Cyclic AMP abhi kya karega? Cyclic AMP is going to activate what? Anybody in the group can tell me what? It is going to activate what? It is going to activate cyclic AMP dependent protein kinase. PK लिख दे, right? Earlier it was inactive. This is active. When we write it, usually बहुत लंबा time लगता है, but when you will do the revision, it is very easier and simpler. It will look very simple. ठीक है? अभी तक क्या देखा हमने? एपिनेफ्रिन आया, ट्रिगर किया उसने एपिनेफ्रिन और ग्लूकागॉन रिसेप्टर्स को, एडिनाइलेट साइक्लेज एक्टिवेट हो गया, इट स्टार्टेड मेकिंग साइक्लिक एएमपी. साइक्लिक एएमपी ने साइक्लिक एएमपी डिपेंडेंट प्रोटीन काइनेज को activate किया. It activated that protein kinase, and protein kinase function is what? It is going to do the phosphorylation. The name is such because it is dependent upon the cyclic AMP, but this is going to do the phosphorylation. phosphorylation glucagon में what we want? We want phosphorylase enzyme to be active. This phosphorylase enzymes become active, and since it is active, we call this as activated form, and this gets phosphorylated, right? So remember, phosphorylated form is active. Earlier it was phosphorylase B, that is inactive form. So कैसे याद रखेंगे? A is active, and this is going to do what? It is going to start breaking the glycogen down. And what do we know about the structure of glycogen? Ye is another question. Glycogen is what? It is a complex monosaccharide. So it has an inner core. Inner core is what? Protein. And what is that protein? That protein is glycogenin. ठीक है? Glycogenin is a protein that is also known as primer. Right? It gets first glucose molecule, and that is donated by UDP glucose. So who donates? That is another question. Who donates the glucose for the synthesis of glycogen? UDP glucose does. You have to remember, uridine diphosphate is the donor, and from where it came? It came from UTP. Right? UTP comes from one side, and the glucose 1-phosphate comes from the another side. There is enzyme, right, which helps in mating of UTP and glucose 1-phosphate, and that UDP glucose is synthesized, plus 1 PPI is being released. Though pyrophosphate means 2 phosphate, 1 phosphate from here and 1 phosphate from glucose 1-phosphate. They come together, and that is being released. ठीक है? Glucose 1-phosphate कहाँ से आया था? Glucose 1-phosphate is released by the breakdown, right? When are we doing the breakdown? We are doing the synthesis. This is coming from glucose 6-phosphate. You have to always make sure what we are talking about, right? And that is coming from glucose. Now we are doing the synthesis. हम breakdown का बात नहीं कर रहे हैं. We are doing the synthesis, and that UDP glucose will be attached to this protein glycogenin. Now another question is, this glycogenin is a protein, and that is also known as a primer. UDP glucose. UDP is the donor of the glucose that helps in the synthesis or building up the glycogen. Now which amino acid is involved? There is this trip, right, in the glycogenin. Uske corner pe, if you imagine, this is a protein, and that has this end, right? This is going to help in the synthesis of the glycogen. कितने आएंगे? 1, 2, 3 up to 11 glucose molecules are going to come in a line. They will hold hands into hand. These are your hands, and these hands are what kind of bond? Another asked question in the exam. You have to label it sometimes. That is 1,4 glycosidic linkage. They are 1,4 glycosidic bonds. They have it. If it is beyond 11, let's say 17, 18 हो गए glucose molecules, right? So around 11, a SNP would be made, ठीक है? और जो rest जो 5-6 glucose molecules हैं, they will create a branch point. And that branch point is 1,6 glucosidic linkage. Remember, and that's how you will keep on building the glycogen. Ultimately कैसा लगेगा glycogen? Inner core is protein. Now there are so many branches. Branches ka, you have the sub branches. So I can say, if when I move towards the periphery, the branching increases. So when you do the glycogenolysis, from where it will start breaking it apart? क्या सब जगह से you are gonna break it apart from here? No, it has only one reducing end. That is a funny thing. ठीक है? So यहाँ से शुरू होगा. Then this will go away, this will go away. This slowly it is going. You have to melt it down by just removing one glucose molecule at a time, right? That is how we will start from periphery and we will move towards the inner core, right? And we know the deep branching enzyme is going to remove this 1,6 linkage. Now, another important thing here is whenever one glucose is removed, that glucose is removed in the form of glucose 1 phosphate normally. 1,4 linkage may, when you are going to rip one glucose, that is glucose 1 phosphate. But when you remove the branch point, then that glucose is simply in the form of glucose. So approximately 10% is free glucose molecule. That is how we get garbad in the energetics also. Okay, so you have to know when you remove the branch point, that branch point glucose is released in the form of glucose, and to utilize it you have to convert it into glucose 6 phosphate by the enzyme, and that enzyme, this step is energy requiring step. This step is energy requiring step that utilizes 1 ATP that we have studied in glycolysis also. Okay, this is about how we regulate the glycogen synthesis and glycogenolysis. Do you have any questions? I have many other questions coming up. If you have anything, you can just type it in the chat box. Okay, and now when we talk about the muscular glycogenolysis, right, so many questions were from this, so I clubbed everything together. We know we have white muscle fibers, and then we have a red muscle fiber, right? In the white muscle fibers, usually there is a fast twitch, and then when there is a fast twitch, there is an incomplete oxidation of glucose, and the glucose will be converted into lactate, right? In the red one, there is slow twitch, right? And the glucose is oxidized completely. Oxidized. Good. We know this for the fasting state. Fasting state में who is going to contribute as a food, like for the survival of the organs? RBC. RBC is dependent upon glucose all the times. Maybe it is a fed or fasting state. What about the brain? We have two vital organs, brain and the heart. Like most important priority ho jata in the starvation state to take care of these. Brain is dependent upon glucose. In the fed state, in the fasting state, in the prolonged fasting state, 2/3 of it can utilize the ketone bodies. 2/3 R is the ketone bodies. Rest still is the glucose. So in the prolonged fasting state, it utilizes both. Heart is mainly dependent upon the fatty acid, right? Then why it has GLUT4? Why the heart has GLUT4 when it is dependent upon the fatty acids? See, GLUT4 is used when there is like baby is not born yet, it's still in the fetus. That point in time, the fetus utilizes glucose via GLUT4. Another is during the myocardial infarction, right? It is unable to utilize fatty acids. It needs like energy fatafat se chahiye, and that during that time it makes use of the GLUT4 transporters that are present on the heart walls, right? So you should be knowing these two, three things very, very, very well. Okay. Now when we are talking about GLUT4 again, which of the GLUT transporter is insulin dependent? कौन सा है insulin dependent? You have GLUT1, 1, 2, 3, 4, 5. 4 and 5 are very special. 4 is insulin dependent. Four is insulin dependent. And 5 is for 5. For is fructose. It takes care of the fructose. You should be knowing those two also, right? GLUT1, GLUT2, GLUT3, they work all the same way. But GLUT4 and GLUT5, 4 is insulin dependent, right? Those were pretty much questions I had for today's session. If you have any question, I'll wait for 1-2 minutes. Otherwise, we will close this session. Are there any questions? Okay. There is confusion about the purine and the pyrimidine's metabolism. And majorly commonly asked question is the uric acid level. Majorly asked question is the uric acid level. Do you know when, in under what circumstances, या in which questions, in which under what topics you see the uric acid level बहुत बढ़ गया? कब बढ़ता है uric acid level? When you hear about the Lesch-Nyhan syndrome. ठीक है? When और कब सुनते हो? Hereditary fructose intolerance, galactosemia, right? Lesch-Nyhan syndrome, which enzyme is involved in the Lesch-Nyhan syndrome? HGPRTase, ठीक है? Hereditary fructose intolerance, FBI, यहाँ पे वर्ड बताया था मैंने, that is aldolase B. Aldolase A is related to glycolysis. अभी fructose intolerance में you have aldolase B. ठीक है? Then the galactosemia में which enzyme is involved? That is galactose 1 phosphate uridyl transferase. Right? Where else? Lesch-Nyhan syndrome हो गया, hereditary fructose intolerance हो गया, galactosemia हो गया, and also severe alcoholism, right? The severe alcoholism. During that time, there is this question. Somebody asked me, ma'am, lactate का level भी बढ़ जाता है, uric acid का level भी बढ़ जाता है, right? Why the hell the uric acid level is related to the increased lactic acidosis, और lactic acidosis का condition क्यों होता है? See guys, there कैसे होता है ना कि you have to remove the uric acid from the system, and that should be eliminated via the renal tubules. Renal tubules have only one exit gate. ठीक है? एक ही exit gate है. Either uric acid can go out or lactic acid can go out. ठीक है? So when the uric acid levels are up, uric acids are given priority for the exit. That is why lactic acids keep on accumulating in the body, and that is happening usually during the severe exercises, and also the uric acid levels be up hai. ठीक है? So lactic acidosis goes up because the uric acid is given the priority to be eliminated from the body. ठीक है? That is happening during the severe exercises with the patients with the high uric acid levels. ठीक है? Under what, how do we control the uric acid? Which drug is used for the uric acid? One of recall questions, very very important. कोई बताएगा? Hey KJ, not much of a difference you should find in English or the English version of the PG content. You want to study less? That is the least I have given, like very short version I have given in the NEET, this one. And the shortest version would be your LMRP, which you will see soon. And the new questions, most important ones, we have the TND for. ठीक है. Now uric acid का level कैसे कम होता है बताओ? How do we control which drug is given? We give a purine analog. That is a prodrug, allopurinol. We give allopurinol. Allopurinol targets which enzyme? Xanthine oxidase. It targets xanthine oxidase. Very good, Aditya. Do you remember the pathway, entire pathway? It's fun actually. When you see the breakdown, how the things are being broken down. The purines are what? Pure as gold? Let me do it quickly for you guys. Purine. Purines are pure as gold. So we are going to break down AMP and the GMP. They are nucleotide. Nucleotide में क्या होगा? You have the phosphate, you have the sugar, right? If you remove them, then you are left with the base. You have phosphate, you have sugar, you have base. ठीक है? अब you have to remove everything one by one. Let's start with adenine. You remove the phosphate first, and then what you are left with? Phosphate को remove कर देंगे तो what you are left with? You are left with adenosine. You are left with adenosine. That is a nucleoside. ठीक है? Because the phosphate is gone. Now what you do? Adenosine has amino group. So you will remove the amine group. How do you remove the amine group? Very important enzyme, adenosine deaminase. ADA, right? ADA. That is related to a disease, SCID. That is related to severe combined immunodeficiency disease in which the T cells level and the B cells levels are hampered. Very important question. ठीक है? अभी phosphate have removed, you have nucleoside. What would be the next step? What you will remove? उसके बाद you have to remove the sugar. Adenosine में से पहले amino group is removed, then you are left with inosine. Next step is you are gonna remove the ribose 5 phosphate. ठीक है? अब क्या हुआ है? Phosphate भी चला गया है, sugar भी चला गया है, but you are left with base. Base, now this base adenine do not have the amino group. ठीक है? Inosine का क्या बन जाएगा? Inosine would be become hypoxanthine. Right, Ankit? Hypoxanthine. Right? That would be a base. Now, if you will degrade the GMP, same way you will remove the phosphate first, then you are left with guanosine. Right? Guanosine do not have amino group, so deamination का step ही नहीं है. Right? And then you will be left with what? You will be left with guanine once you remove the ribose 5 phosphate. Now, ribose 5 phosphate को remove करना, you are removing a sugar to it, and there is this enzyme known as PNP. PNP removes the ribose phosphate sugar. That is purine. What is the name? That is purine nucleoside phosphorylase. Purine, and for nucleoside phosphorylase. Now here comes the fun part now. Abhi you have guanine, you have hypoxanthine. You have two things, right? And the fate of them is they can be converted into a common thing, xanthine. ठीक है? अब this xanthine has two ways. या तो घर जाएगा या तो ऑफिस जाएगा, right? Office is recycling, working again. That is salvage pathway. Salvage pathway. We can reuse this for the synthesis of the AMP and the GMP. Similar, you can say it कि जैसा breakdown किया वैसा ही हम synthesize करेंगे. And who will be helping it to do it? कौन सा enzyme है? That is your HGPRTase enzyme, and deficiency of this enzyme is related to what? Lesch-Nyhan syndrome. क्या? सेल्फ म्यूटिलिएशन, किडनी स्टोन, इंक्रीज्ड यूरिक एसिड, लेश्नायन सिंड्रोम हो गया. ठीक है, दिस इज salvage pathway. What if you break it down? This xanthine can be broken down by xanthine amino acid, amino oxidase into uric acid, right? Which is inhibited by allopurinol if the uric acid level really goes up, right? So now in just one page, you know so many things. How to break down the purines, ठीक है? You know ADA is related to the SCID, right? You know PNP, who is removing the phosphate group, ribose sugar, right? And you know the xanthine amino oxidase, you know Lesch-Nyhan syndrome. Just one pathway, so many things are there, right? And there are conditions in which PI level goes down. अगर inorganic phosphates का level कम हो जाएगा, then what is going to happen? These monophosphorylase will be accumulated, क्योंकि इनको what they need the phosphate to become the ADP, to become the ATP. ठीक है? If PIs are not available, then what you will be having? Then you will be having increased elevated level of the monophosphates. You will be having increased level of the monophosphates. ये level ज़्यादा हो जाएगा, then what you will see? Then you will again see the uric acid, क्योंकि वो बहुत ज़्यादा, it's gonna be accumulated. Then the system will think, okay, let's break it down. Then the uric acids will be increased. So PI का level we have just seen, uric acid levels are increased under all these conditions. So we can say under all these conditions, the PI phosphate levels are low. Simple as that. ठीक है ना? Like with one thing, you have came to know so many, you can answer so many questions. Just they are how they are interrelated with each other. ठीक है? So I had just these many questions for today, right? If you have more questions you want to ask, just keep telling me, right? My Telegram ID initially I have given you. This is my Telegram ID, or you can just leave a message in the comment box also on these YouTube videos. Most of the times I try to take questions from these also, right? So now you should be able to answer all the questions related to the glycogen. Rate limiting enzymes पता होने चाहिए, who is helping, like who is regulating it. Regulation is very very important, I'm telling you, because if regulation is disturbed, then that is going to cause a disease. And for you as a doctor, you should know what are the underlying mechanism of the disease. So that is biochemistry, and that is the mutations. Balance bigड़ेगा, disease is gonna happen, and you have to cure it. And how you are going to cure it? Just to bring everything in the balance again. ठीक है? Now you know the glycogen, you know purine का metabolism से क्या क्या issues होने वाले हैं. The uric acid level, very very important question, you know, right? The MODI, you know, if you have more questions about these MODI things, right? Diabetics में बहुत सारे questions बनते हैं that are part of a biochemistry also. You can drop me, right? I can help you answer those questions also. But there was one question in this NEET PG exam about the aging. ठीक है? We had lots of like discussions about the aging also, कि telomerase answer होगा कि stress hormones होगा. See guys, देखो, the aging is dependent upon many things. I can tell you that aging में हम लोग बहुत सारी चीज़ें consider करते हैं. Question, first of all we have to figure it out what question is asking you. If the question is asking you the age of the cell, right? Age of the cell is dependent upon the size of the telomere, ठीक है, in our body, and that length of the telomere is maintained by the enzyme telomerase, right? So when the cell is aging, telomerase levels are going down, so telomere length is shortening, it is getting shortened, छोटा होता जाता है. दैट इज टेलिंग यू ओके ठीक है सेल कितने टाइम बाद डेथ हो जाएगी सेल की, दैट इज रिलेटेड टू सेल. Now when we are talking about our aging, right, our aging or aging of the cells and then the aging of the tissues, and we take कि पूरा बॉडी का कैसे हमें पता चलेगा, right, then that depends upon the metabolic process, how fast our metabolic process is. So and the metabolic process actually depends upon kitna hoga wear and tear of the cells. When we breathe in, then we what we produce in our body, we produce the reactive oxygen species. Those, imagine those are like of a corrosion, जो आपकी बॉडी को कोरोड करते हैं, इरोड करते हैं, डिग्रेड करते हैं. ठीक है. सो एनी सच कंडीशंस विच इंक्रीजेस द लेवल ऑफ ROS, reactive oxygen species in your body, right, they are going to decrease the aging, right, वो कम कर देंगे एजिंग को. एंड ROS कब बढ़ेगा? अंडर द स्ट्रेसफुल कंडीशंस. बहुत हीट में हो फॉर द प्रोलॉन्ग पीरियड ऑफ टाइम, बहुत कोल्ड में हो, शॉक लग गया है, right, some kind of UVB radiation has hit in you, right, that increases the all these condition, all these stressful conditions, they increases the your breathing rate, oxygen rate, and that is going to cause the production of the reactive oxygen species, hence thereby decreasing your life. And see, longevity is one thing, right, metabolic process is one thing, programmed cell death is different thing. So you have to keep an eye on the words if they are asking you these kind of a question. Okay, so with this I'll close this. Please thank you very much for listening, and keep asking questions. I'll be really glad if you ask me questions and any kind of a doubt you have, right? I just want you to have